Hey sweetest potatoes, so today we are going to do a slightly different video where you can see my hands. Yeah, so we'll be studying more about the secondary structure of proteins. So you might think that natively, like if all the proteins are made up of the same 20 amino acids, then one day the properties of these proteins be same. And like if you denature or unfold protein, they have similar characters which are averaged to the dangling side chains. But however, if you take the 3D structure, that is the native physiologically folded protein, it is seen that the proteins are specified by their primary structure and they have a unique set of characters. Thus, we can say that the properties of a protein are largely determined by their 3D structure. So in this video, we shall discuss about the structural features of proteins, the forces which hold them together, and the organization of these complex structures, which will also become the basis for understanding the structure, the function, and the relationship which is necessary to understand the biological role of proteins, the dynamic behavior of proteins, and also show how they fold so that to their native structures. So with this intro, let's go into the topic. So we'll be we'll be talking about all these topics, that is mainly in the secondary structure of proteins. We'll be talking about alpha helix, beta-pleated sheet, the differences, and also you have something called as the beta bend or beta turn. So coming to introduction, so what is primary structure of protein? So before going into that, I would like to give you an analogy. So consider this to be the primary structure of proteins, and the proteins are made up of, and all these beads are like the amino acids. The amino acids are attached on the string, the sequence are attached together, and this kind of forms or looks like the primary structure of protein. So, and each of these beads represent the amino acids which are present. So coming back to this, so what is primary structure? So it is the simplest level of protein structure, and it is a simple sequence of amino acid in a polypeptide chain, which is super similar to this beaded structure. So that is referred to as the primary structure of protein. Now coming to the secondary structure of protein. So what is secondary structure? So the conformation of polypeptide chain by twisting and folding is referred to as the secondary structure. And here you can see the amino acids are very close to each other in a sequence. And for the proteins, it means the specification of a regular polypeptide bond by folding them into helices and pleated sheets and turns. So when a lot of these structures, they get folded and you have another structure. So this is, you can consider this as the secondary structure of proteins, where you can see a lot of twisted turns, loops, helices and stuff like that. And also I'm showing off my hand knitting skills. Yeah, so this kind of forms like the secondary structure of protein, and all the sequences are very close to each other. And then you have, and finally what happens, the native form or the physiological structure of protein. So you have many folded proteins like this, they combine to form a structure like this. So there are almost like 3 sheets of that of this in this. So this kind of forms like the physiological. This is an analogy to make you understand. So if it's folded, for example, there are 4 polypeptide chains in hemoglobin, so that forms the the tertiary structure or the native form of the protein. So yeah, with this we'll go forward with the topic. And before going into the alpha helix, beta sheet and beta turn, which are the different secondary structure of proteins, we have to know a little bit about what are the helices. So what is helical structure? So helices are the most striking element of the protein secondary structure, and if a polypeptide chain is twisted by the same amount of each of its alpha carbon atom, it is assumed to have this structure, that is the helical structure. So just a preview. So what do you mean by this alpha carbon? So alpha carbon, it refers to the first carbon which is attached to the functional group, like a carbonyl group or any functional group. So that carbon is referred to as alpha carbon. And if there is the same amount of alpha carbons in a polypeptide chain, so they tend to have this, twisted kind of appearance. And like we have already learned about these angles, that is the phi as well as psi angles. uh, in a helix, they're specified by these angles also, that is the phi and psi. If you have not seen the Ramachandran video, you can check that out in the same channel. So, and here you can see something called the N and P, right? So here N, it refers to the number of polypeptide units per turn. So for example, if you see here, you can see that here you can see N is equal to 4, which means that there are 4 polypeptide units. You can count the polypeptide units which are given in a black dot. So there are 4 polypeptide units, hence N is 4. And P is the P refers to the distance of the helix rise along the axis per turn. So here P is 0, and here you can see so this is the distance between this helical rise is referred to as P. Okay. And you also know, the helix can be right-handed or left-handed helix. So for this, to explain this, I actually needed my hands. Okay, so if you consider, so this is your hand, right? So if you consider this as your right hand, and so this thumb, it actually points towards the direction of the helix. So this is the direction of the helix, and the fingers which curl around this right hand, this points along the axis, like the helix axis. You can see the coiling of the polypeptide chain. And how are these fingers glued together? So these fingers are glued together by hydrogen bonds. So you have, these are the different polypeptide chain. This is the direction, and this is the right-handed helix because it's in the right-handed direction. And then you have these polypeptide chains which are joined together by hydrogen bond. So the hydrogen bond acts like a glue. So with that introduction, so let's go into the topic. So alpha helix. So alpha helix was first elucidated by Linus Pauling and Robert in the year 1954, and it is the backbone of a polypeptide. If the backbone is twisted by equal amount of alpha carbons, then it forms this the coil structure, which is referred to as alpha helix. And this helix, so there are some specific angles which you have to remember. That is, the phi angle is minus 57 degrees, the psi is minus 47 degrees, and N, that is the number of polypeptides per turn, is 3.6 residues per turn. And the pitch, that is the P, pitch refers to this distance, so that is 5.4 Armstrong. And if it is that is for right-handed helix, and for left-handed helix, again there are slight variations where only the angles from minus it becomes plus 57 and plus 47, and N is again same, which is 3.6 residues, and pitch minus 3.6 residues, whereas the pitch is 5.4 Armstrong. And the right-handed helix, like it turns. If you take this as a right-handed helix, so the helix turn is in the direction. So this direction, the helix turn will be in the direction of the fingers, and the thumb points towards the helix rise. And you have the hydrogen bond. So the distance between the hydrogen as well as the nitrogen to oxygen bond, that is the NO. So the NO distance is 2.8 Armstrong. And they also have a axial distance between the adjacent amino acids, which is 1.5 Armstrong, and it gives about 3.6 amino acids per turn. See, per turn of this helix, you find about 3.6 amino acids are found. And you also have the hydrogen bond of alpha helix, which are arranged in such a way that the NH bond of the nth residue is towards the COO group, and and this results in a strong hydrogen bond, and the distance between them is about 2.8 Armstrong. And the alpha helix is tightly packed, that is, the atoms, they have the force of attraction here is the van der Waals contact. And you can see in the next so here it's a formation of the alpha helix. So this is the distance which I told you, that is 2 point the hydrogen bond as well as the oxygen bond here. So the distance is 2.8 Armstrong, and you have 3.6 amino acids per turn, and this is the the distance between the helix, between the adjacent amino acid is 1.5 Armstrong. So this here you can see a space filling model of the alpha helix of this is whale myoglobin, and here see the green color gives the carbon atoms, and blue nitrogen atoms is given by the blue color, and you have red for oxygen atoms, and hydrogen is given in H. So this is the alpha helix model of the whale sperm, and this is referred to as the space filling model. Now coming to the examples of the alpha helix. So again you have the alpha helix is found in both fibrous as well as globular proteins, and in case of fibrous proteins they are found in tropomyosin, and also in your hair, nails and stuff. And in globular proteins you can find alpha helices in hemoglobin as well as myoglobin. And chymotrypsin is one enzyme which do not have this alpha helix structure. So now coming to the next one, which is the beta-pleated sheets. And one more thing is that the proline, hydroxyproline and glycine. So what happens is that if the glycine will disrupt the the next one is the beta-pleated sheet. So this beta-pleated sheets were also described by Pauling and Corey in the year 1951. And as with alpha helix, the beta sheet also have repeated phi as well as psi angles, which are allowed in the which fall in the allowed region of the Ramachandran plot. And it also uses the full hydrogen bonding capacity. So the full hydrogen bonding capacity is used of the polypeptide backbone. So in beta-pleated sheets, how you see is that the hydrogen bonding, it occurs between the neighboring polypeptide chain than within one, as seen in alpha helix. So in alpha helix, what you saw is that if you consider this to be the alpha helix, okay, so the hydrogen bond were between the same chain like this. But when you consider this beta-pleated sheet, it is not within one chain, but it is with the neighboring polypeptide chain. So the hydrogen bonding is different in case of the beta-pleated sheets. And these beta-pleated sheets are of two varieties. One is called as the anti-parallel beta-pleated sheet, as well as the parallel beta-pleated sheets. So coming to parallel beta-pleated sheets, as you can see in this diagram here, so the hydrogen bonds are extended in the same direction of the N and the C terminals. So you can see this is the N and the C terminal. So the N and the N and the C terminal, they are in the same direction of the two polypeptide bonds, and this forms the parallel beta-pleated sheets. And also if you consider the angles, the psi and the phi angle, it is equal to plus or minus 180 degrees. And just to have a recap, what is phi angle is the bond which is formed between the nitrogen and also the alpha carbon, whereas psi is the angle between the alpha carbon and the carbon bond, and when a CO and NH bond is referred to as the omega angle. So this is just a recap. So, and then the beta sheet also forms the structural motifs in case of globular proteins. They contain about 2 to 22 polypeptide strands. Now so you have so this is another diagram where you can actually see the hydrogen bonds in the parallel beta sheeted. So you can see the N and the C terminal, and this is how the bonding takes place in case of parallel beta sheet. The main thing you have to remember is the N and the C terminal are in the same direction. So now coming to the anti-parallel beta-pleated sheets. Here you can see the neighboring hydrogen bonded polypeptide, they run in opposite direction. So here you have the N and the C terminal in one direction, whereas the neighboring one, the N and the C terminal, are in the opposite direction. And in the previous one, they are stabilized by, so in case of this, the previous one, that is the parallel beta sheet, they are stabilized by intra-chain hydrogen bonding. So this is referred to as the intra, between the chains, so intra-chain hydrogen bonding. Whereas when you come to anti-parallel, it is interchain. Interchain hydrogen bonding can be seen. So this is another diagram where you can see the N and the C terminal are in the opposite direction, and this type of hydrogen bonding is referred to as interchain hydrogen bonding, which can be seen. So again, when it comes to this beta-pleated sheets, they're found in globular proteins. Usually they have about 2 to 22 polypeptide in a strand, and up to 15 residues long you can see beta sheets, and an average of 6 residues of length 21 angstroms are seen. And 7 standard antiparallel beta sheet, it occurs in a protein which is called as the Jack bean protein of conavalin A. So in that, this is just an example where you can see like 7 antiparallel beta sheets occur. And yeah, these are the occurrence. It is found in structural as well as fibrous proteins, and silk fibrin is the best example. It's also found in amyloid human tissue. So this is like the, this gives you the differences between the alpha as well as beta pleated sheet, where you can see this is coil, here you have a fully extended, and also the about the number of hydrogen bonds. So that can be better explained with this diagram. So here you can see the differences between the alpha helix as well as the beta pleated sheet. So in alpha helix, this is the hydrogen bond is within the same helix, whereas here it is between the between 2 polypeptide chains, and you can see the hydrogen bonds are parallel to the polypeptide. Here the hydrogen bonds are perpendicular to the polypeptide, and these are the differences. That is the axial distance between amino acids is 3.5. Here it is only 1.5, and only one polypeptide chain is there. Here you can have more than one or more constituent polypeptide chains. So the hydrogen bonding, you can see the difference, how they have been bonded. And now coming to the last part of this video, that is the third structure which we are going to study, that is referred to as the beta bend, beta turn, or it's also called as the reverse turn. So the beta bend, beta turn, or hairpin or reverse turn refers to a segment of the polypeptide which will abruptly reverse its direction, and it oftenly connects to the adjacent antiparallel beta strands, and hence they are also called as the beta turn. And I hope you can see this diagram. Once again, yeah. So coming to this non-repetitive structure, so this comes under the non-repetitive structure. So these in case of globular proteins, you can see an average of 31% of them is made up of alpha helix, and 28% is made up of beta sheet, and the remaining polypeptide chain, it is said to have this coiled or looped structure. So the these non-repetitive, this alpha helix and beta sheets are repetitive structures, But these non-repetitive secondary structures, these are very less ordered and ordered than these alpha helix and beta sheet, and they're like more irregular and they're difficult to describe. And the globular proteins, they consist of approximately straight runs of secondary structure which are joined by these polypeptides which runs in the opposite direction. So if you have like 2 alpha helix or many beta sheets, so the beta turn or these non-repetitive structure, they form a link in connecting those two, the alpha helix or the beta sheets. So such reverse turns, beta bends, beta turns are named because it connects the antiparallel beta sheets. And usually all these, the beta bends, they have about 4 amino acid sequence. Usually, like you see 4 amino acid residues are arranged, and they are arranged in 2 ways. So again, these beta sheets are also, even though it's difficult to describe, again there are 2 ways in which the beta bends, or not beta sheets, the beta bends or the beta turns can be formed. And why they're called beta turns? Because these mainly it will go and join antiparallel beta sheets, hence the name. So these are the 2 ways in which the beta bends you can find. So they're referred to as type 1 beta bend and type 2 beta bend. So you might be wondering, what is the difference? It looks so similar, but here if you see, you can see there is a slight change. You can see the red one is here inside, where here it's outside. So that is the only difference you can see between the type 1 and type 2. And there are also a little bit of change in the phi as well as the psi angles. So these are the 2 types of the beta bend. So they differ in 180 degree. You can see this 180 degree flip in the peptide linking the second and the third residue. So the type 1 beta sheet is disordered at a section of the 310 helix, whereas the type 2 beta sheet, it bends at an oxygen atom of residue 2 towards the CP of residue 3, which is usually a glycine residue. So here this is the change what you can see. And now you might be wondering, what is the function of these beta sheets? So the main function of these proteins are that these segments, they have functional role. That is, what they do is they help in binding specific molecules, so that they may be disordered in one protein and they'll be ordered in the other. And also because of all these beta turns, the flexibility of the protein increases and it connects the proteins. It acts like a connecting medium. So only because of this, it is very important for the proper functioning of the protein. So with this, we conclude this chapter, and thank you so much for listening. And yeah, stay tuned for more awesome videos. Thank you so much. And if you like this video, consider subscribing.